Hello, welcome to some video notes. Today we're going to be looking at part 3 of the blood units called division, and it's actually we're going to divide this into 2 parts. So this is part A, and we'll look at part B in another set of notes. And so in today's lesson, we're going to be focusing on cells themselves. So up to this point, we've talked a lot about systems, and then we learned a lot about different types of tissues, and we learned about what cells are like how they can be designed to do specific jobs, because we learned about your blood and why your blood is so important, and what the role that your blood and your blood cells are designed to do. And now we're going to look at them specifically. What is a cell? What are the different types of cells? And what are the things that make a cell a living thing? What are the parts of a cell that build it? Obviously, you're built out of 17 to 20 trillion cells, right? But then what is a cell itself being made out of? What is the thing that you're made of itself? So let's go through this in our hospital. So if you remember here, we're working with Dr. Wong, and here we're looking at another patient has come in. So we've just seen Mr. Patel here, and his mouth has a serious problem. We can see there's some damage on the inside, there's some bleeding, and so we're gonna have to do some investigation as to what is this damage that's happening inside of Mr. Patel's mouth, right? So Nurse Percy did a biopsy, which basically means that she took a tissue sample. So she went into his mouth and they just cut a little bit away from the troubled area, cut a little bit of the sample away. Because what we want to do is spread these cells out onto a slide and see if there's anything wrong with the cells, physically wrong with the cells. So just like you saw with our blood sample, where we had the blood cells kind of all separated out so that we could see them on a microscope, right? We're gonna do that with the cheek cells. We're going to color them a little bit so it's easier to see them, and then we're going to spread them out on a slide so that under a microscope they're much easier to see, so we can determine some properties of these cells and if maybe there's something wrong with them. Okay, so let's go down to pathology and let's do this. Let's look at these cells and see if there's something wrong with Mr. Patel's mouth, right? So thanks. Let's look at this straight away. Down at the pathology, we have another doctor who's specializing in this type of stuff, right? And he's like, you're new, so why don't you learn a little bit about how we do this, right? So we could actually look at some of our own cells rather than just looking at Mr. Patel's, and we could compare them. Maybe there's a good way to understand the differences between what happened to Mr. Patel versus a normal, healthy person, right? That doesn't need to go to the hospital. So we could take a sample of your cells, similar to the way that we take a sample from Mr. Patel. So here you have in your packet on page 15 would be the steps for how you would do that. So you would take a nice, clean, sterile swab, right? It kind of looks like a big Q-tip. And you would rub it on the inside of your cheeks, up and down, back and forth, just like you're brushing your teeth, but you're brushing the inside of your mouth. And because the inside of your mouth, those skin cells that are the cheek cells that make up the inside of your mouth, they can come off pretty easily, right? They're not permanently attached to bone like muscle cells are. They're just kind of a loose covering for the area that is your mouth. So when you rub the inside of your mouth, you will get some of your cells to kind of break away and fall off. So some of those cells will end up on the swab. So then we take the swab and we just roll it on the surface of a glass slide so that the cells will transfer from the swab onto the glass. And then we're going to add a special dye called methylene blue. And methylene blue is going to make it much easier to see the cells. It's going to kind of give a coloration to the fluid, which will make it easier to see the inside of the cells and the outside of the cells. And then it's also going to react very well with DNA, with the genetic information that's inside of the cells. So it will bind to the nucleus, which we're going to learn about today, of the cell, and make it easier to see the nucleus of the cell. So just in general, using this colored dye will make seeing the cells much easier under a microscope. So then once we've added the 1 drop of methyl blue, we put our cover slip on, and then we're ready to go, right? We make sure there aren't any air bubbles and things like that. And then we would look at this under a microscope. Now, obviously, if you're doing this online, you didn't get to do this activity, so you didn't get to see what your cheek cells might look like under a microscope. But we can, again, give you an example of what some of these slides would look like under a microscope for you to be diagramming and answering some of the questions. So here we go. If you were to do this and then look under our microscopes, again, this is magnified a little bit more than what we have at our school. They would actually be a little bit smaller than this, but yes, this is what it would look like. And so you can see that the methyl blue does make it much easier to see the cells. You can kind of see the outside versus the inside of the cell. You can see the circular part that is the cell and the open space that's where they're not, right? And then you can see some sections where there are very, very dark circles inside the center of those cells or some part of those cells. Those very, very dark centers, those are the nucleus. That's where all of the genetic information, or what we call DNA, is located inside of the cell. And we know this because the methyl blue is particularly good at sticking to the DNA. And so it collects in an area where there is DNA, like the nucleus. And so you can see that your cells are relatively large, right? These are much larger than your red blood cells. They're very thin, they're very flat, right? Because they're going to make a tissue layer. So they want to kind of make them very flat, not have them be round or stretchy or things like that. And they are relatively normal size, right? There seems like some of them might be kind of weird looking because in the transfer they got smeared or they got crushed. But the ones that are nice and flat, like the ones that are located here towards the middle, or the ones up here in the corner, in the very top left corner, there's a whole bunch of really good skin cells all located next to each other. And so the skin cells are relatively good size. The nucleus are relatively good size, and the skin cells are just kind of slightly overlapping, but mostly just touching each other in order to form that layer of tissues. So you want to pause the video. You can kind of draw this example of a tissue cell under a microscope, of your cheek cells under the microscope on page 15. And then we'll be able to move forward and we'll look at how these cells compared to Mr. Patel's. Okay, so you did that. All right, so while we were doing this, they did their pathology tests and they made their slides. They looked at Mr. Patel's cells. And unfortunately, there is some bad news for Mr. Patel. His cheek cells are not necessarily looking as healthy compared to your cheek cells. And so if you're looking at here to zoom in on just a set of cells, there's the two cheek cells from an image, right? Those would be normal cheek cells, pretty large, flat, small nucleus, you know. These are what Mr. Patel's cells would look like. All right, and so there are some big differences between his cheek cells versus your cheek cells. First off, there is the number of them, right? So when we do a swab like this, we should get a good number of cells. We should get hundreds, maybe even 1,000 cells over the course of the slide, and which should spread out fairly well. However, with Mr. Patel's sample, there's maybe millions of cells, hundreds of thousands to millions of cells coming away when we do this biopsy. So there are a lot more skin cells in his mouth than there probably should be. There's more cells present than there normally should, because when we're spreading them out over the slide, they're still really close together. They're not really kind of separating so we can see them, and that's probably because there's so many of them. Then you can also see here the inside of his cells. You see those really large purple spheres, right, that take up like most of the cell space? That's his nuclei. So his nucleuses are really, really big, right? So nucleus, they can be different shapes depending on which type of cell we're dealing with. But if your cheek cells have a relatively small nucleus, it's a small part of the cell, and his cheek cells have a really, really big nucleus, right? That's going to be indicating that there's something wrong with his cells. Possibly, since the genetic information is in the nucleus, maybe there's something wrong with his cells. Maybe they're doing too much activity, or the genetic information is being used too much, right? And so what we're seeing here is normal cells compared to his cells and Mr. Patel's cells. And I want you to just take a quick couple of notes about how they are different from each other in your note packets. Okay, so you did that? Good. Okay, so that's on page 15, that you're adding those notes. Okay, so now let's learn some more information about what these cells are, why there are differences between his cells versus your cells, and what are these different parts of the cell, what defines a cell versus what is not a cell. Okay, so let's look at just a simple idea of what a cell actually is. And so here we've got the single cell that we've got from our cheek, right? So this is our purplish cell shape, and then you can see the dark purple color for the nucleus inside. But we want to simplify it a little bit for diagramming. Diagramming is a really useful skill in science because it can take something that's relatively complex under a microscope and we kind of simplify it down to just the general shape and size of the thing that we're talking about. And so there's going to be space for you to be doing some diagramming today. All right, so you're going to be doing a diagram of an animal cell, and then later we'll be looking at diagrams of a plant cell. And so do your best to try to keep it accurate but simple. Don't go overly complicated with the details. Here we've got a really good example. This is a very simple diagram of the cheek cell, right? His cheek cell is flat, kind of boxy in shape, or our cheek cell is kind of flat and boxy in shape. So we made a kind of flat, boxy shape for our cell. And then we've got a purple nucleus on the inside, and then here we have a purple circle to represent that nucleus on the inside, and trying to keep the size of the cell versus the nucleus about the same. All right, so then what we want to do on page 16 is start off with this basic diagram. So there's a spot there for animal cell. Make sure you do this under an animal cell because we're animals. So this is the type of cells that animals have. And then we're going to add some more information to it. And I want you to add that information. And then towards the bottom of page 16, there's some space for you to kind of write out descriptions or write out some information about what these things are. So our first thing that we're looking at is the cell membrane. And so for your diagram, this is the single box shape that you make, right? That single line that you're drawing with your pencil, making this box shape, right? The cell membrane is very important because the cell membrane, it kind of acts like the gate of our school. It is defining what is the inside and what is the outside of the cell, right? And it's there to protect and give shape to the cell and kind of, allow the control of things that go into the cell and things that will leave the cell. Just like our school. We have a gate that goes around. We have a fence system that goes all the way around the property of the school. So you know that if you're on our school property, if you're on the inside of the fence, or you're not on the school property, if you're on the outside of the fence, right? And then we have guards and we have a gate that controls when people can come into the school and when they have to leave the school, right? And so that is kind of like how a cell membrane works, right? The cell membrane kind of defines the shape of the cell, what's the inside, what's the outside, and then it helps control what things can and cannot go into the cell. That's the very first thing that a cell has to have. It has to have the ability to have physical shape, right? It makes the physical shape of the cell, and then control what can go into the cell and what can leave the cell. For example, we want to have glucose and oxygen getting into our cells, but then they want to get rid of waste material like urea and carbon dioxide. All right, so then your single outside layer, that would be your cell membrane. Then in the middle, we've already said this is the nucleus, right? So make sure you're labeling this on your diagram. The nucleus contains genes, which are sections of DNA or pieces of information that give a cell very specific instructions on how to do something. Genes are also what give you your properties, right? So if you have brown hair, you have a gene inside of your cells, inside of the nucleus of your cells, that gives you brown hair. If you have blue eyes, you have a gene inside of the nucleus of your cells, right? That tells them to make the chemicals that give you blue eyes, right? So your height, to a certain extent, your weight, whether you're a boy or a girl, all that information is going to be stored in your nucleus, inside your DNA, the genetic information that we keep inside of our nucleus. So just draw a little circle there in the middle. You can shade it in, if it helps know the difference between different things in the cell. And then make sure that you wrote down on page 16 what a nucleus is. Okay, what is it for? And then finally, for animal cells, our animal cells, very, very simple at this point. There are a lot of things, other things. We'll get in. We're going to do very simple stuff at the beginning. The empty space in the middle, so that yellow coloration that they've put on the inside of the cell for our diagram, or the purplish coloration that kind of fills up the entire center of the cell inside of the cheek cell that we have in our image, that is called cytoplasm. Okay, cytoplasm. So plasm is referring to like a liquid, right? So if you guys remember that our blood plasma, right? That's another word that's similar. Plasma is the liquidy part of our blood. So plasm is referring to liquid. Cyto is referring to a cell. So cytoplasm means the liquid inside of a cell, right? And so it's just this chemical, it's this liquid, it's full of material, so it's not really watery. It's actually kind of jelly-like. It's thicker than just regular water. And it's got lots of stuff inside of it. It's got lots of different materials that will do different chemical reactions. It's got different nutrients. It's got fats in there. It's got glucose in there. It's got salts in there. And it's also kind of just doing all these chemical reactions to keep the cell alive. So a lot of the activity that happens inside of a cell that keeps it alive is going to happen inside the cytoplasm. And the cytoplasm really takes up most of the inside of a cell. It's the most of the empty space that all the other things are located inside of our cell. So what you need to do, make sure you've been adding to the stuff. If you haven't, you need to pause the video before you move on. Finish making your simple diagram on page 16, and then also labeling it, the membrane, the nucleus, and the cytoplasm, right? And then on the pages, on page 16, you have some sections. There's the words, nucleus, cell membrane, cytoplasm, and there's some lines there. And I want you to just write out what it is that these things do. What is their job for the cell? Okay, so you did that. All right, so moving on. So that's our animal cell. It's our basic animal cell, right? Make sure you add those definitions onto page 16 as well. So if we look at Mr. Patel's cells versus your cells, we notice that his cytoplasm is much smaller, right? His nucleuses are much, much larger, and he seems that his cells are much smaller, right? So they're not growing to the full size before they seem to be going through cellular division, before they seem to be making copies of themselves again. So this is a sign that Mr. Patel has cancer, right? So you may have heard of cancer before. Cancer is a problem inside of our DNA in which the cells will start to reproduce very, very quickly. So when a cell divides, a single cell can grow and then divide in two, and it becomes two cells. And then those cells can grow and divide in two, and then the two cells become 4 cells. And then those 4 cells can become 8 cells, and those 8 cells can become 16 cells. You get this idea, right? And so that's really important. We want cells to be able to do this because as you grow, you need to have more cells. Right now, if you're about 17 trillion cells, because you're really small, as you get older and you're like your full height, your full age in your 20s, you might be around 28 or 30 trillion cells, right? So you need a lot of cells to keep growing. So this is an important feature of cells, the fact that they can divide and make more of themselves. Also, it's important because when you get injured, if you fall down, you scrape your knee, right? You've damaged your body, you damaged your skin, you might have even damaged some of the deeper layer of tissue if it's a very bad cut. So you need to repair that. And to repair that, you need to make new cells. And so by reproducing, by making new cells, that's how your body heals itself. Unfortunately, sometimes the cells growing can get out of control, and they can keep growing over and over and over again. They don't stop when they're supposed to. And that creates cancer. So cancer is this uncontrolled growth of cells. They just keep getting bigger and bigger and bigger, eventually creating something we call a tumor, which is a mass of cells taking up space inside of your body. So that's what we found from Mr. Patel. His cells have come from a tumor that must be growing inside of his mouth, okay? So tumors, things like cancer, if we really want to see them very, very clearly, so we have an idea that this might be cancer, right, from the microscope, the light microscope with our slides. But if we want to be really, really sure, we want to get a closer look, right? And so if we want to look even closer at our cells, we want to get really detailed images of a cell, we cannot use a light microscope. There are limits to how much light can make something that's really small look bigger. However, we could use something that's much more powerful than a light microscope called an electron microscope. And so an electron microscope uses electrons, or the electricity, right, that moves through your phone, right? Electrons are the movement, the electricity is the movement of electrons. And you can use those electrons in order to use a special computer program that can get a very detailed image of something that's very, very tiny, okay? And so there are different types. And so one we could use is called a scanning electron microscope, or SEM. And so a scanning electron microscope does what it says. It scans the outside of something. So it looks for very, very high details on the outside of a cell, not looking at the inside of the cell. So for here, we're looking at red blood cells under a light microscope. They're very small, right? And we can get a general idea of their shape and their color, right, and their size. But we can't get a lot of detail about a red blood cell from a light microscope. Here is what it looks like under a scanning electron microscope. Look how much closer we can get of these red blood cells, right? And remember the fact that they are biconcave, they have those dents on both sides of the cell. It's much easier to see those dents looking through a scanning electron microscope than through a light microscope, scanning electron microscope versus a light microscope. So electron microscopes, very, very important for us to really confirm things about cells, because we can get much better images of the really, really tiny parts of them, right? So you can see the differences right away here, right? Okay, so SEM images show the whole surface of these cells, right? And they can show us all sorts, and there's other types of microscopes as well that can allow us to look on the inside of these red blood cells. So if we want to look on the inside of the cancer cells to get more information about them, if we want to look on the outside of the cancer cells to get more information about them, these scanning electron microscopes, or just electron microscopes in general, can really help us out with this. So if we wanted to look at more of the cancer cells here using a scanning electron microscope, we can compare the cancerous cells versus like a normal cell. So we've got a normal red blood cell here, and then we've got a cancerous cell here. And so the danger with cancerous cells is that they can spread. They can continue to grow, and they can spread to different parts of his body. Now that we've identified that Mr. Patel has cancer in his mouth, they're going to have to probably do a quick little surgery to cut out all of the cancerous cells from his mouth so that they don't continue to spread over to different parts of his body. But you can look at the difference here of a cancer cell versus a red blood cell. It's got all these irregular shapes on it. It's much larger and spread out in different directions because it's keeping to increase in size and growing kind of irregular, right? So we can kind of get a much better view of something that it is cancer and confirm that it's cancer by looking at it in more detail using an electron microscope, okay? So also with electron microscopes, we can confirm other parts of the cell that we know exist, and we can look at them a lot easier. So with a light microscope, you can't really see more than the cytoplasm, the cell membrane, and the nucleus of an animal cell. But there are more things inside of our animal cells than that. There are a bunch of other parts that are important, but we're not going to be able to see them through a light microscope because they're actually really, really small. So they're in the cytoplasm. They're floating around inside the cytoplasm, just like the nucleus is, but they're very, very tiny. And so to see them, we would have to use another type of electron microscope. So here we're using a tunneling electron microscope, so it will, or a TEM, and it will allow us to see the inside of a cell. And so here we're looking actually at, this is an example of a plant cell, but the same thing applies. So we can see the barriers of the cell, the inside and the outside, the very, very dark lines, right? The cytoplasm, instead of it being kind of like just a general empty jelly, you can see that it's full of lots of other stuff. All those little gray lines are other parts of the cell that do something else. And then you have the nucleus, right? In the very beginning, in the very center there, you've got that big dark center where the DNA is being kept. And if we zoom in on one little part of the cytoplasm, we can see the mitochondria. And you guys see here with our purple label, that one little bean shape here in the image. And actually there's a bunch of mitochondria. There's one there over on the right side. There's a whole bunch of them over here on the left side, right? So remember, this is three-dimensional image, or it's a two-dimensional image of a three-dimensional thing. So they're all gonna look slightly different from each other because of the way the image is taken. But you can see that they're kind of a bean shape, and they've got a bunch of membranes on the inside. They've got a bunch of folds and lines on the inside. That's the mitochondria. Mitochondria are super important. Every single cell needs to have a mitochondria. All animal cells have mitochondria. All plant cells have mitochondria. Because the mitochondria are what's going to do that respiration that we talked about. Remember the characteristics of a living thing, right? Mrs. Gren. One of the R's was respiration. And respiration is the ability to break chemical bonds, to break things down and get energy out of that breaking of things, right? And so what are we gonna do? We're gonna break down glucose, which is a really good source of energy. And the mitochondria are going to do that. So the mitochondria are actually the part of the cell that breaks glucose into small parts, right? Using oxygen, that's why oxygen is so important, and gets a lot of energy for us. So the mitochondria, you've probably heard this already on the internet, the idea that it is the powerhouse of the cell. That's not maybe the most accurate way to describe them, but it does work, right? That is where a lot of the energy that we need is going to come from. It's going to come from our mitochondria, okay? So using the microscopes, right, at any time if you want to see more things about our cells. As he says, come back if you want to investigate more. So make sure on page 16 you're adding that little piece of information about the mitochondria, because we're doing our definitions, right? Okay, let me add that stuff in. All right, so we've caught up to this point. We've done our animal cells. So then let's move on and see what other information we can learn before this lesson ends. All right, so later that day, after we've dealt with Mr. Patel, we go back, we do some of our paperwork. So much paperwork that doctors have to do. And our nurse that is helping us out says, hey, you're here late. And she's looking over at the aquarium. Right, and he said, Mr. Cooper, by the way, said that there could be more plants in the tank, right? It would have, if it had a brighter light. So basically he said that, you know, your aquarium's looking not so great. Maybe you need some better light in order to help the plants that grow inside the aquarium. And that's this idea, like, you know, that plants need sunlight, they need to do photosynthesis, and sunlight helps them with photosynthesis. So if you're gonna have some plants inside of your aquarium, or you're gonna have aquatic plants, you need to make sure that the light is bright enough so that they can do enough photosynthesis. So the nurse is like, okay, well, we'll have to help out the plants and probably have a more powerful light. That makes sense. We've learned about photosynthesis. We understand how more light can probably help the plants grow a little bit better. But now we're kind of wondering, what do these look like, right? So plants are very different from humans and very different from animals in general. Animals, we have muscles, we have bones, right? We have a respiratory system, we have the lungs, you know, stomach. We've got all these different complicated cells in order for us to be able to move and capture things and eat them and run away from things. Plants are not like that, right? Plants are very slow. They can move, they have movement, they can move and grow in different directions, but they're much, much slower. They don't have, you know, a lot of defined body parts. They've got the roots, they have the stem, all right, they have the branches, they have the leaves, right? They have a very kind of simpler form, you would say, compared to something like a cheetah or an elephant. So let's take a sample of these plant cells and let's look at them and see if there are some parts to these plant cells that are different from our animal cells. What are the things that make plant cells different from animal cells? So if we looked at the plant cells under a microscope, it would look kind of like this, right? And so right away, you're noticing some differences. First off, there's a lot of green coloration inside your plants, right? And so you would probably remember that plants do photosynthesis. And so to do photosynthesis, they need a very special coloring pigment called chlorophyll. And chlorophyll is really good at absorbing light, but it doesn't absorb green light. So green light doesn't get used. So if something doesn't get used, it gets pushed away, it becomes waste, right? So the green light gets released from the plant, and so that's why they look green to us. The light coming off of a plant, when light hits it, is the green coloration. So we see that they're very, very green, right? We can see that they've all got these little green sacs on the inside, and there seems to be a lot of green coloration there. We also can see that there are very, very thick barriers between the cells. There are actually a large distance between the inside of this cell and the inside of this cell, and that's called a cell wall. And a cell wall is a very important structure. It keeps the cells nice and strong so that they can grow against the force of gravity. If you remember, plants do not have bones, right? So the reason why you can stand up, the reason why you can move around and and go up something or support yourself against something is you have bones to hold up your body, right? Plants don't have that. So if plants are going to grow upwards towards the sky as gravity pulls them down, they're going to have something to help, you know, grow them nice and strong. So we have a cell wall, or they have a cell wall between all of their cells that kind of helps give them support and structure, right? It's the idea of thinking of building a building out of bricks, right? If I stack all of the bricks on top of each other, you know, we can kind of grow upwards, but there's a chance that they could fall over because they're not really connected to each other, right? However, when we do build with bricks, normally we put some cement between the bricks, you know, that kind of like grayish coloration between each of the red bricks. And that cement helps hold them all together and starts giving the building better support and better structure. So you can think of the cell wall kind of like that. Okay, so what we're gonna do is we're gonna look at a symbol diagram of a plant cell, and we're gonna add some details to page 16. Okay, so here we've already got our simple animal cell, right? So you've already done a good amount of the animal cell. We're gonna add in mitochondria in just a second though. So your animal cell diagram on page 16 should look something like the one that's over there. And then our plant cell, all right? So we're gonna add the plant cell now. If you don't have the plant cell, you haven't done the plant cell yet, so you're gonna add that in. So you can pause the video, right? And you can kind of add the plant diagram the way that it looks here, right? So try to get all the different layers. The different colorations are trying to help pointing out the different layers as well. And then for the animal cell, make sure you add in the mitochondria, which are those tiny little brownish ones that are in the diagram. Okay, so you unpause the video, right? Because you've added in the details to these diagrams that you need, right? And so now we're going to label them and add our detailed information about the parts of the cell to page 16. Okay, so first off, right, we're also going to compare them as we go through this. Okay, So first we have the nucleus. We already did that, right? So make sure you label the nucleus of the plant cell diagram because plant cells have a nucleus as well. Plant cells have DNA, just like we have DNA. They need information to know how to live, just like we need our information to know how to live, right? They have a cell membrane, just like we have the cell membrane on the outside. They have a cell membrane on the outside, right? They need to control what goes in and what goes out of their own cells, just like we do. So they have a cell membrane as well. And you can probably tell already that they also have mitochondria, right? Even though they do photosynthesis to make glucose, they still need to break it down to get energy out of it. So they do need mitochondria, just like we do. So you need to add in the mitochondria to your animal diagram if you didn't do already. And then you have your mitochondria in your plant diagram, and then make sure that you labeled both of them, that they are the mitochondria. And then we see this cytoplasm, right? So the cytoplasm is most of the empty space on the inside of the animal cell. And just like that for a plant cell, a lot of the empty space, not most of the empty space, but a lot of the empty space inside of a plant cell is the cytoplasm as well. Okay, so you should be good up to this point. These are the things that the plant cells have, and the animal cells and plant cells have in common. Animal cells and plant cells, they have a nucleus, they have a cell membrane, they have mitochondria, and they have cytoplasm. Now, as we move forward, we're going to point out three really important parts of the plant cell that are only present in the plant cell and are not present in animal cells. So plants have these features, but we do not. Okay, we ready? So next, the chloroplasts. So the green colorations, and it would be good, by the way, to use green for the chloroplasts. You can use other colors for the other parts, that's okay. But use green when you're labeling the chloroplasts because they are green, right? Because they're able to do photosynthesis, and the green coloration is why they're able to do photosynthesis. So the chloroplasts, again, plants have the ability to do photosynthesis. We do not. We do not get energy from sunlight that way. Then we also have the cell wall, which is very thick, protective layer that's on the outside of the plant cell. Remember, that gives it the strength to grow up against the force of gravity, right? It makes it so that we can have really tall plants. We have trees, that go up for many meters up into the air and dozens of meters up into the air, right? Because they have the cell wall to hold them all together and to give them strength. And then if you notice in the plant diagram, there is a large empty space in the middle. And that's not really empty, it's actually full of water. And so that is what we call the large central vacuole, or we're gonna say vacuole just for short for right now. So the vacuole is this massive, massive storage of water inside of the plant cells. In fact, about 90% of the space inside of a plant cell will be taken up by water inside this vacuole. And this is needed because plants need a lot of water, right? So they're constantly using water for different chemical reactions, but they're also constantly using water for photosynthesis. So they have the ability to store a large amount of water inside of themselves. And then the water also helps give the plant shape against the weight of the cell wall. The cell wall is very, very dense. It also makes it a little bit heavy, right? So it pushes in a little bit, right? It kind of like, because of the force of gravity, it doesn't really hold the best shape, right? So by having lots of water inside of the vacuole, that kind of creates like a big water balloon on the inside of the plant. And so then that makes water pressure that pushes against the sides of the cell wall and kind of helps make sure that we have the right shape of our plant. And this happens because when you see a plant that doesn't have enough water, what does it look like, right? It wilts. It starts to bend down. It doesn't stand up straight. Seems like it's tired. It just kind of flops over. That's how you know a plant doesn't have enough water and that you need to be watering it. Because if it doesn't have enough water, the cell wall isn't going to be pushed against very well by the vacuole, and so the plant starts to get kind of heavy and just kind of falls over because of gravity. All right, so that's really what the vacuole is for. Okay, so make sure you complete these diagrams, right? And then we will look at page 16, and I will show you what you should have written down if you didn't write it down already for the definitions of the parts. All right, so make sure you get your diagrams and you label them before you move on. Okay. So you've done your diagrams. You can try your best at defining the terms. All right. And so then let's look at these parts and finish up our comparisons of these two cells. Okay. So going through definitions of all the different parts, remember that a nucleus, that is what controls the cell and what holds the DNA, all the genetic information for the cell is there. Cell membrane, that gives shape to the cell. It is the barrier between the inside and the outside of the cell. It also controls what goes in and what goes out. Cytoplasm is the liquid or the jelly-like liquid in all of the other parts of the cell. It's also where all the chemical reactions happen. It's where lots of the active parts of the cell's life will happen. Mitochondria does respiration. It makes energy, also known as ATP, if you really want to know how we call energy, inside of the cell, right? It uses glucose to make energy. Chloroplast does photosynthesis, which makes the nutrients for plants. Remember, they don't need to eat things. They can just make their own nutrition using photosynthesis. The vacuole stores large amounts of material, particularly water, and helps move that material around inside the cell. Right? Our cell wall protects and gives support to our plant cells. And then the last things we want you to be able to do is compare both the similarities and the differences between an animal cell and a plant cell. So you haven't done this already, fill this section out. How are they similar? And then how are they different? Okay, so going through the similarities. Obviously, I got differences first. So I'll tell you the similarities if they don't show up. So similarities between animal cells and plant cells is that they both have a cytoplasm, a nucleus, mitochondria, and a cell membrane, right? So they both have those same three parts, okay? Differences would be animal cells versus plant cells. Animal cells have no chloroplasts, plant cells do. So there we go. Sorry, it showed up in the wrong order. Okay, so there's your similarities. Our differences, no chloroplasts versus having chloroplasts. Animal cells do not have a cell wall, where plant cells do have a cell wall. All right, small vacuoles inside animal cells versus large vacuoles inside of plant cells. There are really, really tiny vacuoles inside of animal cells, but they're very, very temporary. They're not like a big storage area like plant cells do. So animal cells can make vacuoles, but they're very small and they do not last very long. Where a plant cell has a very, very big one. You saw the huge space inside the diagram that's used for the vacuole. And then if you want to think about the shape, typically animal cells are much rounder in shape, where plant cells are very, very boxy and square in shape. Okay, so if you need more time to write this stuff down, go ahead, you can pause the video. If you need to go back and review something, go ahead as well. But if not, then that is it up to this point. Oh, sorry, forgot about this. One more thing, I forgot about this part, sorry. Real quick before we finish, just a real quick test for your ability. A, B, C, D, E, and F, I want you to say if they're animal cell or a plant cell. So look at the image, all right, do your best to determine, do you think it's an animal cell or a plant cell based on the properties inside the image. Okay, did you do it? Give this a shot. Okay, A, animal cell, right? No cell wall, no chloroplasts, right? So those are the good, no central vacuoles. Those are the things that make it easier to look to see if we're looking at a plant cell. B, all that green means it's got to be a plant cell, right? Because it's got all the chloroplast inside. C, right? We really don't see much inside. Actually, that looks like adipose tissue. That's fat tissue inside of an animal. There really isn't much inside a fat tissue except for fat. But there's no cell wall. There's no chloroplast or anything like that. D is actually a plant cell. Now, you're thinking that you don't see any chloroplasts, and that's true because what you're looking at is the root part of the plant, not the leaf part of the plant. The roots, which grow underground, do not have chloroplasts because there's no sunlight underground, so there's no reason for them to have chloroplasts because they can't do photosynthesis. But they do have a cell wall, right? You can see this really thick barrier between the parts of the cell. That's their cell wall, so that means it's a plant. E, that is an animal cell, that is actually an amoeba, right? It's got a big flexible membrane, and it's got all these different compartments on the inside, but it does not have a cell wall. And then F, we would also be looking at another plant. And again, it's actually another type of root cell. Again, you can see the nucleus, you can see these really, really thick cell walls between each of the cells. So that tells you that is a plant cell. So some of these were a little tricky, but hopefully you can see now why they are an animal cell or a plant cell. Okay, so that is it for these notes. And if you have any questions, please let me know.